Hi everyone. So let us study human embryology together. I have personally found out that human embryology is best studied if you read it like a textbook. And the reason why I say that is because there's going to be a lot of in-depth knowledge where few pointers in PowerPoint is not going to help you. Yeah? Alright, so let's start, okay? So human embryology is a study of how a human embryo develops from a fertilized egg into a fetus, right? So remember, fertilized egg into a fetus, right? Involves division, it involves cell division and differentiation. So fertilized egg, fetus, division, differentiation, which are the processes that give embryo its form. Right, so stages of human embryology. First is germinal stage. The stage begins with fertilization and continues until the embryo implants into the uterus, right? Which is usually 1 or 2 weeks. Embryogenesis. The stage lasts for first 8 weeks, right? So number 3 to 8. During this time, the embryo develops from a single cell into a multi-layered fetus with rudimentary organs, the primary organs, yeah? Then we have prenatal development. The stage includes pre-embryonic, embryonic, and fetal development. All right. All right, so let's use some mnemonics here, okay? So germinal stage is first 2 weeks. Remember the mnemonic zygote, great journey. All right, so Z, zygote forms. G, growth through rapid cell division, right? We said embryogenesis. It is about, germinal stage has cell division and differentiation, right? J is journey to the uterus for implantation. So we have zygote's great journey from the minute zygote forms through fertilization to journey to the uterus for implantation. All right. All right. So we'll study further that it gets implanted in the ampulla, etc., etc. Okay. Embryogenesis usually weeks 3 to 8. Embryo begins to take shape, right? So you actually have a seed, and now the seed is growing, right? Blastocyst develops. T is 3 layers: ectoderm, mesoderm, endoderm, right? And S is structures form, heart, limbs, organs, right? So we just learned that rudimentary organs are going to start forming, right? Prenatal development is weeks 9 to birth. Fetal growth roadmap. Remember that. You just remember this little baby. First is this zygote's great journey. Embryo begins to take shape. Fetal growth is a little cute little baby. All right, so growth is growth and refinement, right? So we already had rudimentary organs in embryogenesis, right? Now we are going to refine it. It's like we have already something that's revised, it's in draft, and now we are going to release it, right? We are ready to publish something, right? So rapid brain development, yeah? And O is organ systems mature. We already had rudimentary organs. Now you're not going to be born with something that's rudimentary. You want something that's well refined, perfect, and that happens during the prenatal development, right? So prenatal development is weeks 9 to birth, yeah? All right, so organ system mature, womb-based movement begins, and the baby starts moving, yeah? So T is third trimester. It's crucial for survival outside. So third trimester is extremely important for the survival outside when the baby comes out, yeah? All right, so early development stages. All right, fertilization is a process. The first is fertilization, yeah? Fertilization is a process in which a sperm cell and an egg cell join to create a zygote, which then develops into an embryo, right? So fertilization is when a sperm cell and egg cell join to create a zygote. So first is zygote. We just learned, right? It is also known as syngamy, generative fertilization, or impregnation, right? Steps of fertilization: acrosome reaction. The sperm's acrosome release enzyme to help it penetrate the zona pellucida, right? So first of all, what the sperm does, sperm's acrosome release enzymes to penetrate zona pellucida. Okay, so I'm coming in, move everybody, so that's where the acrosomal reaction takes place. Zona pellucida, penetration. All right, so we had a reaction, now I have to penetrate, yeah? So sperm penetrates the zona pellucida. Right, the third is sperm egg attachment. The sperm egg gets attached. You open something, you're going in, now you're attaching yourself, right? Excuse me. The sperm attaches to the egg. The membrane fusion, the sperm and egg membranes fuse, all right? So first, acrosomal reaction, there was a reaction. Second, there was penetration. Third, there was an attachment, okay? And then we had membrane fusion, right? To fuse something, you have to attach something, right? You have to connect something. So that's what happened. Fertilization in humans. In humans, fertilization usually takes place in the fallopian tube. It's usually, most of the time, it's an ampulla. The zygote travels down the fallopian tubes and becomes a morula, right? So zygote travels in the fallopian tube and it becomes a morula, yeah? So morula reaches the uterus and becomes a blastocyst. The blastocyst implant in the uterine wall. All right. Well, first, sperm makes contact with the egg. Second, acrosome reacts with the zona pellucida. Here I am. I'm coming. Be careful. All right. The third is acrosome reacts with perivitelline space. There is a perivitelline space and acrosome reacts with it. What does it use? It uses enzymes. We need enzymes, yeah? All right. Plasma membrane of sperm and egg fuses, right? So as you can see, the sperm is right here and it's getting fused. All right. So we have a little bit of fusion here. All right, sperm nucleus enters the egg. All right, the sperm nucleus is entering the egg. All right, so cortical granules, number 6, cortical granules fuse with the egg plasma membrane, which renders the vitelline layer impenetrable to the sperm. All right, so we can see here that's the sperm nucleus is taking it off, saying, I'm coming. All right, so this is a vitelline layer. This is egg cytoplasm, yeah, usually jelly-like floating. This is the perivitelline space, right? We saw that it was coming in, and as you can see, there's an egg. There's a hydrolytic enzymes, right here, trying to enter. It's egg plasma membrane. This is cortical granule, right? And this is sperm nucleus taking it off and coming in. All right, and slowly there's going to be a fusion, okay? All right. These are the protein receptors. So enzyme is going to react with the protein receptors, right? So it's not just the enzyme, but there is someone there to receive the sperm. Come on in, you know, like that. All right, so journey of an egg. The journey of an egg from ovulation through fertilization and implantation is a complex process. It involves release of egg, fertilization, attachment, fertilized egg to the uterine lining. Ovulation, excuse me. An ovary releases a mature egg. Ovulation usually occurs around day 14 of a menstrual cycle. So when you get your periods on the day 14 from your periods, ovulation usually occurs, right? And that's when you start getting all the feels. All right, fertilization. A sperm fertilizes the egg within the 24 hours of ovulation. Fertilization occurs one of the fallopian tubes. A fertilized egg is called a zygote, right? A sperm fertilizes the egg within 24 hours of ovulation. Okay, fertilization occurs in one of the fallopian tubes. A fertilized egg is called a zygote. So the minute there is a fusion of sperm and egg, it's called as a zygote, yeah? So it usually takes 24 hours of ovulation. That's all you need, okay? So when you go outside, you're dating around, be careful, all right? Okay, so implantation. The zygote travels down to the fallopian tube and to the uterus, right? So that's how it implants, all right? Implantation happens because zygote is, going down to the fallopian tube into the uterus. The zygote attaches to the uterine lining, right? It's right on the uterine lining. The process called implantation. Implantation usually occurs about 6 days after fertilization. All right, so remember number 6, implantation marks the beginning of the pregnancy. So implantation usually happens about 6 days after fertilization. And what does the zygote do? The zygote travels down and implants itself, right? So it's like you put a seed, and there's a special place where you put a seed, okay? You don't put on top of the soil. There has to be 1.5 inch in a perfect line, and then the seed grows into a fully grown plant, all right? What happens if the fertilization implantation does not occur? If fertilization does not occur, the endometrial lining is shed during menstruation. If implantation doesn't occur, the fertilized egg passes out the body during menstruation. So when you are actually going through menstrual cycle, especially women, you will actually find yourself, looking at some of the eggs that come out. It's gross, but you have to understand that it is equally important to shed your uterine lining as much as it's important to be pregnant, okay? Because we need it, okay? So it needs to be done. All right. Journey of an egg. Fertilization normally occurs in the ampullary region, ampulla of the fallopian tube, probably within 24 hours of ovulation. The two pronuclei grow, move together, and condense in preparation for syngamy. All right, and cleavage in about 24 hours. Cleavage is the start of cell, right? We've seen cleavage, right? Those who know meiosis and mitosis understand what cleavage is. All right, division of the zygote into blastomeres begins approximately 30 hours after fertilization. All right. Uh, Pronuclear fusion as such does not occur, alright? The two pronuclear envelopes disappear and two chromosome groups move together to assume positions. You know, there's ready, alright? Now we have chromosomes, we actually have genes coming up. On the first cleavage spindle, no true zygote containing a membrane-bound nucleus is formed, right? First, okay, we have an ovum, we have fertilization, acrosomal reaction, right? We have fusion of the plasma membrane here, fusion of egg and sperm. Pronuclei, we have two, as you can see, a pronuclei, two. It's not just one, two, and the two fuse and they form a zygote. We have two cells, right? From the two cells, what do we have? We have four cells. And from the four cell, we have eight. And then we have morula, right? And then we have blastocyst, right? So the minute it gets implanted, as you can see, it's journeyed through the fallopian tube. And now, blastocyst is right here. It is implanted in the uterine wall. So it traveled, right? There's actually a place, right? So it finds the right place. An unfertilized human secondary oocyte surrounded by the pellucida. First polar body can be seen, spermatozoa can be seen outside the zona pellucida. All right. Fertilized human oocyte before fusion of the pronucleus. Two polar bodies can be seen beneath the zona pellucida. All right. So you can actually see the two polar bodies, right? All right, so let's start. Journey of an egg. We just described the journey of an egg, right? Step 1, egg leaves the ovary and enters a fallopian tube right here. Step 2, sperm enters egg and then unites with the nucleus, right? Step 3, fertilized egg divides. So we just saw the cell division, right? 4 cells, 8 cells, uterus, and there you go. Your cells attached to the uterus, right? And here you have blastocyst, right? All right, fertilization, cleavage, compaction, differentiation. All right, we just said, you know, cell division and differentiation are the bases. Okay. All right. We also have cavitation, right? As you can see, there's cavity here. Inner cell mass, blastocyst cavity. There's also trophoblast, right? Day 6, zona hatching, right? Implantation, uterine epithelium. We just said, you know, blastocyst connects itself, attaches itself to the uterine lining. All right, that's where it's parked. That's it. You're going to be parked there for 9 months. All right, epiblast. This is epiblast. Epi is usually on top of something. Hypoblast is, hypo is usually below something that's not also below epi. And then we have tropho. All right, so below hypo, we have tropho, right? Remember truffles. You know how truffles are layered, right? So remember that, and you'll remember, you know, the cell mass differentiation, right? Okay, bilaminar disc. Oh, so this is a bilaminar disc. As you can see, there are two. A bilaminar disc formation. Mesoderm formation after day 12. We have, so you understand, by day 23, we are usually having umbilical cord. So we actually have an organ that's developing, right? All right, so mesoderm formation. As you can see, mesoderm forms. This is the mesoderm, right here. And you can actually see a little bit of pinch right here too. Right, so that is the primitive streak. Okay, mesoderm spreading. So this is amniotic sac. All right, this is exocoelom. This is a chorion. This pink lining around is a chorion. This here is trophoectoderm, right? So you understand the trophoblast, right? This is the ectoderm, and now here is a trophoectoderm, so it mixed. Okay, so that's mesoderm. Again, this is a yolk sac. All right, so this lining here, the pinky one, is chorion. This is the amnion, right? This is the embryo. As you can see, there's already embryo. This is the digestive tract. So we actually have a digestive tract day 23. Why? The embryo needs food to survive, right? Amniotic sac enlargement. Your amniotic sac enlarges. Umbilical cord is right here because it needs food. And how does it get food? It gets from the mother, right? So we actually have umbilical cord, which is actually connected to the digestive tract, and this is where the embryo is. A zygote cleavage is the process of rapid mitotic cell division, right, that occurs after fertilization. So we actually had meiosis and then we have mitosis, right? So mitotic cell division that occurs after fertilization when a zygote divides into multiple cells to form an embryo. So the zygote is a single cell that divides into smaller cells called blastomeres. I remember this, raspberry. The cleavage process doesn't cause the zygote to grow, so the resulting blastomeres are the same size as the original zygote, right? The cleavage process continues until a hollow sphere of blastomeres called blastula forms. In mammals, the blastula develops into a blastocyst. For us all mammals, the blastula develops into a blastocyst. This is zygote. A cleavage is 8-cell cleavage, blastula cross section, right? As you can see, gastrulation, we have blastophore. We have a hollow region. Gastrula cross section. So as you can see, there's already a blastopore, right? So you can see a little bit of space. Cleavage consists of repeated mitotic division of the zygote, resulting in rapid increase in number of cells. These embryonic cells become smaller with each successive cleavage. So the size is not increasing. The cells are becoming smaller, but the whole cleavage of the zygote, it actually has the same shape as the zygote. It has multiple cells in it, that's all. Cleavage occurs as the zygote passes along the uterine tube towards the uterus. During the cleavage, the zygote is in zona pellucida, so it still has zona pellucida around it. Division of zygote into blastomeres begins approximately 30 hours after fertilization. Subsequent cleavage divisions follow one another, forming progressively smaller blastomeres, right? So 2-cell, 4-cell, 8-cell, morula, the size is not changing. The cell cycle is quite long. The first 2 cell cycles begin being around 24 hours each, thereafter reducing to 12 to 8 hours. So it picks up speed, but it takes a little time. Subsequent cleavage, progressively even smaller blastomeres, right? Cell division is asynchronous. Daughter cells may retain a cytoplasmic link through much of the immediately subsequent cell cycle. We have mid-body as a result of delayed completion of cytokinases, right? No centrioles are present until 16 to 32 cells are seen, but amorphous pericentriolar material is present and serves to organize the mitotic spindles. All cleavage divisions after fertilization are dependent upon continuing protein synthesis, which up to 8 cell stage is derived from available maternal mRNA, right? And thereafter from embryonic mRNA, right? So the embryo has its own mRNA, but that initially it's usually through maternal RNA, and that is until 8-cell stage is achieved, right? We saw the 8-cell, then multiple cells, and then it implants, right? Hence, initial cell divisions are independent of mRNA synthesis. So before 8 cells, the cell divisions are independent of mRNA synthesis. Early cleavage up to formation of 8 cell requires pyruvate or lactate as metabolic substrates, but thereafter more glucose is metabolized and may be required. Initially it's pyruvate or lactate, eventually it's glucose, right? Up to the formation of 8 cells, cells are essentially spherical, touch each other loosely and have no specialized intercellular junctions, right? So up to 8 cells, cells are essentially spherical, touch each other loosely, there's no connection, and have no specialized intercellular junctions. After 9 cell stage, the blastomeres change their shape and tightly align themselves against each other to form a compact ball of cells called as compaction, right? So slowly and gradually they're becoming more sophisticated. They're not just loose. They're taking shape. They're getting ready to be a full-blown embryo, right? Cells flatten on each other to maximize intercellular contact, initiate the formation of gap and focal tight junctions. So they're flattening. Okay, so whenever you have to have a compaction, you cannot be uneven on all the sides. You have to flatten and you have to start connecting, right? As a result of process of compaction, the embryo forms a primitive protoepithelial cyst, which consists of 8 polarized cells in which the apices face outward and basolateral surfaces face internally. So, protoepithelial cyst. So 8 polarized cells in which the apices face outward and basolateral surfaces face internal. So basolateral surfaces, base lateral surface on the both sides, they are facing internal, but they usually have protrusion on the outer external side. As you can see, this is a compacted embryo. This is a fully expanded embryo. They came together, they connected. This one looks spherical, this one looks flattened, and as you can see, they're apices. These are apices, but on the bottom you can see it's, you know, basolateral and it's flattened. Obviously, you're on a surface, you have to flatten yourself. You can't have apices on both the sides. Yeah. Morula, 2 cell population are formed in the 16-cell embryo that differ in phenotype, polar and apolar, right? One that's important, one that's not important. It's just steady right there. And position, superficial, deep, right? The outer polar cells contribute largely to the trophoectoderm. We just saw there was a formation of trophoectoderm, yeah. Whereas the inner apolar cells contribute almost exclusively to inner cell mass in most embryos. Generation of cell diversity to either trophoectoderm or inner cell mass occurs at the time 4th-5th day in the 16-cell morula. We can actually see cell diversity beginning, right? During the next cell division, 16 to 32 cells, a proportion of polar cell again divide differentiatively as in the previous cycle, each yielding 1 polar and 1 apolar progeny, right? So morula stage embryo is number day 4. As you can see, we have a fully formed morula between 16 to 30 cells. Proportion of polar cells again divide differentiatively as in the previous cycle, each yielding 1 polar and 1 apolar progeny. So we actually not only have 8 cells, from 8 cells went to 16, it started differentiation again, 16 to 32 cells. We can actually see a cell, first we had cell division, cleavage, and now we have cell division on a very higher level. Blastocyst. Blastocyst is a very early stage of development when the zygote becomes a ball of cells with a fluid-filled center and an inner mass of cells, right? So zygote, cleavage, blastula, cross-section, gastrulation, blastocyst, endoderm, ectoderm, blastopore, right? This is gastrula, right? Gastrulation is a very important part of embryology, right? And so anyone ever ask you, what is the most important part of your life? Say gastrulation. That's going to be the most important part, okay? So blastocyst is a very early stage of development when the zygote becomes a ball. So from a zygote, you're going to a ball of cells with a fluid-filled center. So there's actually fluid inside. An inner mass of cells, a bunch of mass. It looks like a jelly, yeah? This happens only after a zygote has divided into 36-ish cells to form a morula. So let's say from a zygote, you went to a 36 cell to be a morula, yeah? After division of 32 cells, the outer polar cells complete their differentiation and into a functional epithelium, display structurally complete zonar tight junctions and begin to form desmosomes. All right, so after 32, you are in a morula. Now you're going to start forming tight junctions and begin to form desmosomes. The nascent trophoectoderm engages in vectorial fluid transport from uterine cavity to the inside of the embryo. We did see there's an umbilical cord formation. We saw there's a digestive tract, right? Generate a cavity that expands in size during 32 to 64 cell cycle and converts the ball of cells to morula to a sphere, the blastocyst, right? So we actually have a fluid transportation happening already, right? Once the blastocyst forms, diversification of the trophoectoderm inner cell mass lineages is complete and trophoectoderm differentiative divisions no longer occur, right? So there's actually a stop to the differentiative divisions in trophoectoderm once the blastocyst forms and diversification of the trophoectoderm inner cell mass lineage is complete, okay? All right, so what is blastocyst? Let me move this. So human blastocyst, day 5, zona pellucida, trophoblast, blastocele, inner mass jelly, embryoblast, empty zona pellucida, hatched blastocyst. The blastocyst hatches from its zona pellucida at 6 to 7 days, possibly assisted by an enzyme similar to trypsin. There's an enzyme that helps us hatch, okay? So you understand that enzymes are so important. All right, early pregnancy factor, an immunosuppressant protein, is secreted by trophoblastic cells, and appears in the maternal serum within 24 to 48 hours after fertilization. EPF forms the basis of pregnancy tests during the first 10 days of development. Right, the outer cells of the blastocyst, the trophoblast or the trophoectoderm, are flattened polyhedral, which possess ultrastructural features typical of a transporting epithelium. All right? The trophoblast covering the inner cell mass is the polar trophoblast, and that surrounding the blastocyst cavity is the mural trophoblast. All right. Implantation occurs during a period of 7 to 12 days post ovulation. This is endometrium. This is inner cell mass, embryoblast. This is blastocyst cavity, right? We said there's a cavity. This is outer layer trophoblast. Even at the early stage of cells of the inner cell mass are already arranged into an upper layer closest to the polar trophoblast, the epiblast, which will give rise to embryonic cells, and a lower layer, the hypoblast, which has an extra embryonic fate. Dorsoventral axis of developing embryo and a bilaminar arrangement of the inner cell mass are both established at or before implantation. Okay, so dorsoventral axis, developing embryo and a bilaminar arrangement of inner cell mass are both established at before implantation. So even before the implantation occurs. Attachment to the uterine wall, right? So it has this place we just discussed. The implantation includes the following stages: dissolution of zona pellucida, orientation, adhesion of the blastocyst onto the endometrium, right? We saw an endometrium lining, trophoblastic penetration into the endometrium, migration of the blastocyst into the endometrium. This is actually a process of how it attaches itself to the uterine wall, right? Spread and proliferation of the trophoblast, which involves and specifically disrupts and invades the maternal tissues. The site of implantation is normally the endometrium of the posterior wall of the uterus, nearer to the fundus than to the cervix. It's closer to fundus than to the cervix, right? Site of implantation. And so that's the blastocyst. Inner mass cell. Trophoectoderm. This is trophoectoderm integrins, right? Growth factor, these are cytokines, right? Microvilli on syncytiotrophoblasts, these are microvilli. Why do we need it? We need to attach ourselves, right? So we can't just connect without anything, right? We've seen it before. We've seen that most of the cells usually have some sort of organs to connect itself, right? This is pinopause, this is luminal epithelium, endometrial glands, these are endometrial capillaries. Integrins on receptive endometrium. So we have two anchors to itself, right? So it connects itself. It cannot just connect to a flat surface. We need something to connect, yeah? These are growth factors and these are cytokine, right? Cytokines are very important. I have another video for immunology. If you want to learn, you go ahead and learn. I have talked about cytokines in that video, right? So we'll keep going through the process again and again. So you remember ovulation, right? We have follicular cell. We have fertilization, sperm cell nucleus connects, 2-cell stage, 4-cell stage, 8-cell stage. That is cleavage from 2 to 8. Now we have morula, right? We just discussed 16 to 32 is morula. Early blastocyst, 36-ish plus is early blastocyst. It has inner cell mass, right? Late blastocyst, this is the blastocele cavity. We can actually see the trophoectoderm, trophoblast, and it connects to the endometrial lining right here. It finds its place, it connects, it's closer to the fundus than to the cervix, right? Why do you think that's way? Because it's ready to pop out in 9 months, you know. There you go. Formation of extraembryonic tissues. Epiblast and amniotic cavity. Closest to the implanting phase of the trophoblast. We are talking about the epiblast and amniotic cavity here. A basal lamina surrounds a spherical cluster of epiblast cells and isolates them from all the other cells. Epiblast cells adjacent to the hypoblasts become taller and more columnar than those adjacent to the trophoblast, and this causes the epiblast sphere to become flattened and the center of the sphere to be shifted towards the polar trophoblast. Amniotic fluid accumulates and the amniotic cavity is formed, right? So now we have an amniotic cavity. So what happened here? Okay, so the mechanism is, all right, epiblast and amniotic cavity are closest to the implanting phase of the tropho. So this is the tropho and they're very close to it, right? Basal lamina surrounds a spherical cluster of epiblast cells. So we actually have a basal lamina surrounding the spherical clusters, right? And isolates them from all the other cells. There's a little bit of cell isolation happening here. Epiblast cells adjacent to hypoblast, so these are the epiblast cells and they're adjacent to hypoblast, right? Become taller and more columnar. As you can see, they're taller now and they're more columnar, right? Which actually causes the epiblast sphere to become flattened and the center of the sphere to be shifted towards the polar trophoblast. Slowly and gradually there's a shift that's happening. And you can actually see the amniotic fluid accumulates in the amniotic cavity. So what's actually happening, there's a growth formation happening for a space to be created. So there is a cavity that's created and there's fluid inside. As you can see, there's amniotic ectoderm. So we can actually see there's a little bit of sophistication happening in this particular step. Why? Because there has to be space formation occurring. All right. Hypoblast in the yolk sac. All right, hypoblast is a term used to delineate the lower layer of cells of the early bilaminar. We had epiblast, then we have hypo, then we have tropho, right? During early implantation, the hypo extends beyond the edges of the epi and can now be subdivided into those cells in contact with the epiblast, basal lamina, the visceral hypoblast, and those cells in contact with the mural trophoblast, the parietal hypoblast, right? The parietal hypoblast cell and proliferating and spreading along the mural trophoblast, the cavity that the layer initially surrounds is termed as the primary yolk sac, right? So as you can see, this is the hypoblast. It's in German, hopefully you understand. All right, that is the amnioblast, right? So we can actually see the amniotic cavity that's forming. This is the trophoblast, this is the epiblast, this is epi, right? You can actually see vesicovo ontello. They're talking about amniotic cavity, right? Okay, and this is the cincture trophoblast, and this is the zygote trophoblast, okay? So we can only see parietal hypoblast cells are proliferating and spreading along the mural trophoblast. The cavity that the layer initially surrounds is termed as a primary yolk sac. The cavity is actually called the yolk sac, right? Why? Because there's an extra space that is formed. At the same time, a space appears between parietal hypoblast, uterus membrane, and the mural trophoblast that limits the circumference of the hypoblastic cavity, right? The resultant small cavity inside the lines by hypoblast, right? So we have two cavity formation. Hypoblast is termed as a secondary yolk sac. We have primary yolk sac and we have secondary yolk sac, yeah? All right, so visceral hypoblast cells are believed to be important in many aspects of early specification of cell lines. The cells induce the formation of primitive streak, thus establishing the first axis of the embryonic disc. They are also believed to be necessary for successful induction of the head region and for the successful specification of the primordial germ cells, right? Okay, so as you can see, we have trophoblastic lacunae, we have enlarged blood vessel. Here we have cincture trophoblast. We have cytotrophoblast. You can see multiple cells that are around. Here we have amniotic cavity. Here we have epiblast, right? Columnar. They grew in size, right? And this here is the exocoelomic uterus membrane. It's covering it. This is exocoelomic cavity. This is a primitive yolk sac, yeah? Here you can see the figrum coagulum, okay? So you can see that. Amniotic cavity, this is exocoelomic cavity. This is the primitive yolk sac. Okay, so oropharyngeal membrane. This here is the oropharyngeal membrane. These are primary villi, trophoblastic lacunae. Exothalamic cyst, right? We just said there are two cysts that's formation, two cavities formation. This here is extra embryonic splanchnic mesoderm right here. Extra embryonic cavity, chorionic cavity. This is the extra embryonic somatic mesoderm, chorionic plate. This is the plate. This is the secondary yolk sac, right? So we have one cavity, we have amniotic cavity, and we have secondary yolk sac. This here is a connecting stalk. This is maternal sinusoid. After the formation of the secondary yolk sac, a diverticulum of the visceral hypoblast, the allantois, forms towards one end of the embryonic region, extends into local extra embryonic mesoblast. It passes from the roof of secondary yolk sac to the same plane, right? So we have amnion, we have chorion, we have two amniotic folds. We have folds here. Anterior end of the embryo, this anterior end. This is arcantron, this is allantois, right? This is yolk. This is a yolk sac. It's like a bag, okay? Extra embryonic coelom. This is the extra embryonic coelom. This happens to be a vitelline membrane, okay? Extra embryonic mesoblast. Extra embryonic tissues that encompass all tissues that do not contribute directly to the future body of the definitive embryo, right? So extra embryonic mesoblast, it will come to cover the amnion, secondary yolk sac, and the internal wall of the mural trophoblast, and will form the connecting stalk of the embryo when it's contained allanto-enteric diverticulum, right? So now slowly we are only getting more sophisticated. This is the amniotic cavity. As you can see from this we went to this, right? So we have primitive streak formation, which actually started happening. We just saw. We have mesenchyme, we have notochord right here, right? We have allantois, we have primitive streak right here, we have primitive node, we have epiblast. This is the epiblast. This is the extra embryonic mesoblast, yeah? The first mesoblast extra embryonic layer gives rise to the layer known as extra embryonic mesoblast. So now we have a formation of an extra layer arranged as mesothelium with underlying extra embryonic mesenchymal cells, right? So we have extra embryonic mesenchymal cells. Later, extra embryonic mesoblast population mushroom beneath the cytotrophoblastic cells at the embryonic pole, forming the course of developing violet stems and villi and the angioblastic cell that will give rise to capillaries within them and the earliest blood cells. This here is primary chorionic villus. This part is secondary. This is tertiary. This is villus chorion. This is decidua basalis. These are maternal blood cells. Cytotrophoblastic shell, chorion. This here is extra embryonic mesoderm. This here is cytotrophoblast. And this here is extinctio-trophoblast, right? Initially, the extra embryonic mesoblast connects to the amnion, to the chorion over a wide area. Continued development expansion of extra embryonic coelom means that this attachment becomes increasingly circumvented to a connecting stalk, which is a permanent connection between the future caudal end of the embryonic disc and the chorion. The connecting stalk forms a pathway along which a vascular anastomosis, now we have vasculation happening around the allantois, establish communication with those of chorion, right? So here we have yolk sac, we have amniotic cavity, and this thing is amnion. We actually have a primitive stick that's developing. We also have extra embryonic coelom. This is extra embryonic coelom, right? Connecting stalk. This here is connecting stalk. This happens in 2 weeks, okay? All right. So the vegetal pole of blastula flatens. This is vegetal hemisphere. Here we have blastocele. Some cells change shape and move inward to form arcantron. This is blastopore. Okay, other cells break free, becoming mesenchyme. So some cells become blastopore. Some cells they go free and they become mesenchyme. Thin extension of mesenchymal cell attached to overlying ectoderm, so it connects itself. The arcantron elongates as assisted by a contraction of mesenchymal cells. So the arcantron elongates. Mouth will form where the arcantron meets the ectoderm. The mouth forms. This ectoderm, this is endoderm, this is blastocyst, this little here. This here is now architron, and this is mesenchyme, the cells. This is blastopore, right? Remember the cells, some cells become blastopore, some cells become mesenchyme. This is a blastocyst cavity we've been talking about, yeah. It's ectoderm on the outside, endoderm on the inside. This is architron, all right? Blastopore will form at the anus of the mature animal, mature human. Okay, gastrulation, very important part, yeah. Gastrulation is a critical stage in the development of most animals where a blastula reorganizes into a multi-layered embryo called a gastrula. The multi-layered embryo is a gastrula. So what happens during gastrulation? Cell migration. Okay, cells on the outside of the embryo move inward, reorganize the embryo. From cells outside, go inside. Germ layer formation. 3 primary germ layers, endoderm, mesoderm, and ectoderm are formed. Body axis establishment. Gastrula establishes the dorsal ventral cranial caudal and anterior posterior axes, you know. So you know how we have x and y axis in gastrulation, all the axes. We actually have a framework that's starting. Organogenesis. The germ layers develop into a specific organs, right? So when does gastrulation occur? In humans, gastrulation occurs around 14 to 21 days after fertilization. What is the term gastrula derived from? The term gastrula comes from the Greek word gasta, which means stomach. Yeah. What are some abnormalities that can occur during gastrulation? Heterotaxia, a condition where the heart, lungs, and abdominal organs develop on the opposite side of the body. That can happen, yes. So it can actually occur on the opposite side of the body. If the organ formation is not done right, then it can, you know, form on different places. All right. So in human embryo, after about 3 days, the zygote forms a solid mass of cells by mitotic division called a morula, right? This then changes to a blastocyst consisting of an outer layer called a trophoblast and an inner cell mass called embryoblast, right? So outer layer is trophoblast, inner cell mass is embryoblast. Filled with uterine fluid, the blastocyst breaks onto the zona pellucida, right? Undergoes implantation. Inner cell mass initially has 2 layers, the hypo and the epi. We just saw that. At the end of the 2nd week, a primitive streak appears, right? We saw a primitive streak appeared. The epi in this region moves toward the primitive streak, dives down into it, and forms a new layer called endoderm, pushing the hypoblast out of the way. This goes on to form the amnion. The epiblast keeps moving and forms a 2nd layer, the mesoderm. The top layer is called the ectoderm. Yeah, the top layer is ectoderm, below ectoderm is hypo. Gastrulation occurs in reference to the primary body axis. We just saw was axes that were forming, right? Anterior, posterior, dorsal, ventral, etc., etc. Germ layer formation is linked to primary body axis as well. However, it is less reliant on it than gastrulation is. Okay. This is blastula with blastocyst. This is gastrula. As you can see, gastrula is a much more sophisticated form of blastula. Yeah. Ectoderm, endoderm, architron, blastopore, blastocyst. Yeah. This is blastula with blastocyst only. We have gastrula. So let's say you started your own clinic. Yeah. You're running your own clinic by yourself. You're just a doctor. Now, from just one person, you not only became the head of the clinic, now you have multiple associates that work for you, and you also have an extra space for yourself. Let's say you have a cafeteria where you can actually meet. So that's going to be a blastopore where most of your ideas are going to come into fruition, right? 3 doctors, 4 doctors coming together and saying, well, let's do this, okay? So gastrula, ectoderm, endoderm, architron. Endoderm is one of the germ layers formed during animal embryonic development, right? Humans are animals too, haha. All right, cells migrating inward along the architron form the inner layer of gastrula, which develops into endoderm. The endoderm consists at first of flattened cells. We saw that, right? Something gets flat only to grow, yeah? Which subsequently become columnar. So we saw that they were converted into columnar shape. Okay, it forms the epithelial lining of the whole of the digestive tract, except part of the mouth and the pharynx and the terminal part of the rectum, which are lined by involutions of the ectoderm. It also forms the lining cells of all the glands, which open into the digestive tract. including those of liver, pancreas, and we have liver glands, we have pancreatic glands, yeah. Epithelium of the auditory tube and the tympanic cavity for the ears, the trachea, the bronchi, and the alveoli of the lungs, the bladder, and part of the urethra, and the follicle lining of the thyroid gland and thymus. The endoderm forms the pharynx, the esophagus, the stomach, small intestine, the colon, the liver, the pancreas, the bladder, the epithelial parts, the trachea, bronchi, the lungs, thyroid, and parathyroid. And that's why gastrulation starts with gastro, which means stomach. You know, everything's going to start forming. Okay. For endoderm embryology, this is endoderm, right? As you can see, we have buccopharyngeal membrane right here. We have lung bud, okay? As you can see, there's eyes too. This is a foregut. These are pancreas. This is a midgut. There's a hindgut, all right? You can actually see cloacal membrane. This is allantois. This is vitelline duct. We actually have a gallbladder. We also have liver right here, okay? All right. Mesoderm, during gastrulation, some of the cells migrating inward contribute to mesoderm, right? So some cells are going inside and they are actually contributing towards meso, all right? An additional layer between endo and ecto is meso, it's in the middle. Formation of mesoderm leads to the development of coelom. Organs formed inside a coelom can freely move, grow, and develop independently of the body wall, while fluid cushions protects them from the shocks, right? That's why it's in the middle, it's meso, all right? Mesoderm has several components which develops into tissues: intermediate meso, paraxial meso, lateral plate meso, and chorda meso. The chorda meso develops into a notochord, right? We saw there was a notochord formation. Intermediate mesoderm develops into kidneys and gonads. Paraxial mesoderm develops into cartilage, skeletal muscle, and the dermis. Lateral plate mesoderm develops in circulatory system, including the heart and the spleen, the wall of the gut, and the wall of the human body. So we actually saw the gut's formation, right? So mesoderm is a very important part because, as you can see, you know, a lot of organs are developing. Skeletal muscle is developing. Imagine not having a skeletal muscle. Yeah, imagine not having a dermis. Imagine not having a cartilage, okay? All right? So imagine not having a wall of gut, you know, and imagine not having a heart and a spleen. You know, there's no point in being a human, okay? Through cell signaling cascades and interaction with ectodermal endodermal cell, the mesodermal cells begin the process of differentiation. The mesoderm forms, all right, smooth muscle, bone, cartilage connected to the lymphatic system, dentine of the teeth. Imagine not having dentine of the teeth. Circulatory system. Imagine not having a circulatory system. The genitourinary system, serous membrane, spleen, and notochord. So as you can see, this is intermediate mesoderm. Is a lateral plate of mesoderm, lateral plate on the side, yeah. Paraxial mesoderm. This is the paraxial. This is notochord, the one in the flower, like right in the middle. This is extra embryonic mesoderm. All right, the mesoderm, embryology. This is very important. Intermediate mesoderm, kidney, gonads. Chorda mesoderm, midline mesoderm, notochord, precordial plate, intervertebral disc. Paraxial, somatic mesoderm, somites and head. We have lateral plate mesoderm that gets converted into, that grows into somatic body cavity, pelvis, limb bones, very important. Extraembryonic, splanchnic circular system, right? So from paraxial somatic mesoderm, we have somites, sclerotome cartilage, we have dermal myotome. Yeah, so that's dermis, skeletal muscle, myotome, right? The skeletal muscles. Sclerotome cartilage, vertebrae, ribs, endotome, endothelial cells, dorsal aorta, arthrotome, vertebral joints, yeah. Syndotome, those are tendons. So mesoderm, you know, as you can see, is very important because it gets converted into, it grows into ribs. We have joints, we have endothelial cells, we have tendons, skeletal muscle. So pretty much most of the musculoskeletal system is created, you know, it starts growing during from the mesoderm, so it's very important. The ectoderm generates the outer layer of the embryo and it forms the embryo's epiblast. The ectoderm develops into surface ectoderm and neural crest and the neural tube. So we have neurology development, yeah. We can actually see that a lot of it is going to develop from ectoderm. Surface ectoderm develops into epidermis, hair, nails, lens of the eye, sebaceous glands, right? Cornea, tooth enamel, the epithelium of the mouth and the nose. The neural crest of the ectoderm develops into PNS, peripheral nervous system. Adrenal Central nervous system is brain and your spinal cord. Everything else is peripheral. Medulla, melanocytes, facial cartilage. All right, so very important. Neural tube of the ectoderm develops into brain, spinal cord, posterior pituitary, motor neurons, retina. Yeah. The anterior pituitary develops from the ectoderm tissue of Rathke's pouch. Okay. Anterior pituitary in your brain develops from the ectodermal tissue of the Rathke's pouch. As you can see, there's a lot of development of hair, skin, nails, neural crest, right? We also have neural tube ectoderms in the brain, spinal cord. Yeah. Okay, so we have ectoderm. Outer ectoderm, we have epidermis, hair, nail, sebaceous, olfactory epithelium, your nose, mouth epithelium, lens, cornea. Mouth epithelium, you have anterior pituitary, we have tooth enamel, and cheek epithelium, yeah, cheek cells. Peripheral nervous system, from ectoderm we have neural crest that develop in PNS, we have Schwann cells, neuroglial cells, sympathetic nervous system, we have parasympathetic nervous system, right? We have adrenal medulla, melanocytes, facial cartilage, dentine of the teeth. Neural tube is brain, neural pituitary, spinal cord, motor neurons, and we have retina from neural tube. Neurulation refers to the folding process of vertebrate embryos, which includes the transformation of neural plate into the neural tube. An embryo at this stage is termed the neurula. The process begins when the notochord induces the formation of CNS by signaling the ectoderm germ layer above it to form a thick and flat neural plate. The neural plate folds in upon itself to form neural tube, so it folds it in, right? So it's starting to form a tube, which will later differentiate into spinal cord and the brain, eventually forming the CNS. Very important system, yeah? Different portion of the neural tube formed by two different processes called primary and secondary neurulation in different species. The primary neurulation, the neural plate creases inward until the edges come in contact and fuse, right? So they're actually going inside, all right, and forms a tube. The primary neurulation, the neural plate creases inward until edges come in contact and fuse. They come together, they go inside and come together, yeah? In secondary neurulation, two steps for neurulation, tube forms by hollowing it of the interior of a solid precursor, yeah? All right, so neurulation refers to folding process of vertebrate embryos, which includes the transformation of plate into the tube. So it goes from a neural plate into a tube. All right, so let's find out what's the step, yeah? During the third week after the fertilization, for the development of three types of tissues formed during gastrulation occurs, yeah? Here is the notochord. We saw the typical flower-like object, yeah? Cells within the mesoderm form the notochord, which runs the length of the embryo. Ectoderm folds inward, forming a groove that runs the entire length of the embryo. Neurulation is completed with the formation of neural tube. Somites and the coelom develop from the mesoderm. So we have neural tube, we have two somites, we have coelom, and we have amnion right here. And during neurulation, the germ layers begin to develop into the organs and the tissues of the body. Mechanisms of the development. Cell specialization is the process by which cells develop into specific cell types with distinct functions. This process is also known as cell differentiation. Oocyte and sperm, blastocyst, ectoderm, mesoderm, endoderm, ectoderm, neuron epithelial cells, mesoderm, red blood cells, cardiomyocyte, we saw muscles, right? Endoderm is liver cells and pancreas. Totipotent cell, pluripotent cell, multipotent cell, unipotent cells, yeah? How does cell specialization occur? Gene expression, a specific combination of genes are turned on or off, which dictates how a cell functions. Specification, determination, differentiation, a series of events that occur during embryonic development, yeah? So we have specification, we have determination, and differentiation, which is a series of events that occur during embryonic development. What is cell specialization? Why is it important? Sorry. Embryonic development allows a single fertilized egg to develop into a complex organism. Protects DNA, allows multicellular organisms to express fewer genes in individual cells, which protects genes from damage. Examples of specialized cells. RBC carry oxygen in the body. Sperm cells contain genetic information from the male parent and need to reach the egg cell to fertilize it. Egg cells, reproductive cells in people assigned female at birth. Yeah, we just saw the fusion. All right, so nerve cell transmit information. Muscle cells have components that allow them to complete a specific purpose, right? Picking something up, contracting, stretching, etc. All right, ciliated epithelials move substances in one direction, such as mucus in the airways. So ciliated, we have cilia, yeah? Yeah. All right, so all these cells have 20,000 genes distributed in 46 chromosomes. How specialization is it? Let us take the example of RBC. How does it even occur? How does it happen? All these cells have hemoglobin gene in the nucleus. This gene is not expressed in all cells. It is expressed or turned on only in RBC, where it has a role. So hemoglobin gene, very important for transportation of oxygen, right? Yeah. All right. So it's actually the HB gene has RBC. All right. As you can see, the particular gene is turned on. Yeah. So hemoglobin gene is very important RBC, and it's only present in RBC. It is not present in all the other cells, right? Try finding hemoglobin in leukocytes. Yeah. Specification determination. Cell difference determination is a process by which a cells commit to a specific development pathway and become specialized cells. Consider yourself, you're a pre-med, then you learn the general human system, and you study. Eventually you decided that you want to be a neurologist or you want to be an orthopedic. So how does that determination specification actually happen? Right? It's the same, cell determination. It's a fundamental process for a multicellular organism. Is responsible for many important functions. How it works: cells detect different chemicals in different regions of the embryo. Cells receive inductive signals from nearby cells. Cells follow a specified path towards a cell differentiation. You came into a med school, yeah, and then, you decided, you learned many things, but then you decided, or your friends told you that you're very good with bones. Why don't you try to become an orthopedic, all right? So cells follow a specified path. You said, okay, let me do it. And cells become irreversibly committed to a specific cell fate, all right? If you're an orthopedic, you can't go back to be a primary care. If you want to, you can, but most of the times you're going to be an orthopedic surgeon or specialized with everything bones, okay? So you can't just go back. You can't decide, maybe I should be a neurologist, after having like 5 years of residency. You cannot do that. Your specification and determination is already done, okay? What it's important for: cell determination is essential for normal development. It helps maintain homeostasis or internal equilibrium. It allows for adult tissue to regenerate, yeah? So it's important, yeah? Essential for normal development. Imagine not having orthopedic. Imagine not having any specialization. Imagine not having any specification. You'll just stay as general medicine. Wouldn't you want to be a subject matter expert with highly skilled, masterful knowledge of a specific subject? Yeah, so it's important, yeah? So how it's studied: long read sequencing can help identify structural variants in DNA, alternative splicing events in cell type specific mRNA isoforms. Mathematical modeling can be used to study RNA. How is to study RNA velocity using single cell sequencing data. All right? How it can be influenced. Extracellular environment and the contents of the cell's genome can influence cell determination. So who's actually power of authority? It's actually the cell's genome. Mechanical clues such as hydrostatic pressure can influence cell's fate. How much good you are under pressure can actually, decide which field you want to go to, right? So there's actually hydrostatic pressure, and that is influencing your cell's fate. Are you good under pressure? You can be an emergency medicine. Do you want 9 to 5 easy, breezy type of lifestyle? Go for a specialization that works best for you. All right, so you can see that an extra outside environment is actually deciding what your determination and specification is. The specification determination is a very important part, okay? All right. Cellular differentiation is a biological process in which a less specialized cell develops and matures to possess a rather distinct form and function. It is a process in which a cell changes into another cell type, right? During cell differentiation, changes may include cell shape, cell size, membrane potential, metabolic activities, and responsiveness. These changes are brought about by modification in gene expressions An example of cell differentiation development of a single cell zygote into a multicellular embryo that further develops into a more complex multisystem or distinct cell types of a fetus. Yeah. The cell size, shape, polarity, metabolism, responsiveness to signals change dramatically such that a less specialized cell becomes more specialized and acquires a more specific role. A cell that underwent differentiation is described as differentiated. A differentiated cell means a cell that has changed in a form and mature form being generalized into being more specific in terms of function. The term undifferentiated is used to describe a cell that has not yet acquired a special structure and function. An undifferentiated cell, therefore, would be a primitive cell or a progenitor cell that is yet to undergo cellular differentiation. All right, so an undifferentiated cell can be called as a primitive or a progenitor cell. Stem cell. It is likely that all cells originate from stem cells and acquire their roles as they develop. Stem cell population is often positioned at the top of hierarchical structure that represents cellular composition. A stem cell, as you can see, the stem cells in a tray, they can be neuron, they can be brain, osteocyte, they can be a bone. Enterocytes, entero means intestine, hepato is liver, so hepatocytes, liver cells. Cardiac cells, heart, cardio is heart, right? Osteo is bone. So stem cell, it can be anything, okay? It can do multiple things, all right? Stem cells are usually jack of all. I can do whatever I want to do, okay? The first embryonic cells that arise from the division of zygote are ultimate stem cells. These stem cells are described as totipotent. I'm totipotent, okay? Because they have potential to differentiate into any of the cells needed to enable an organism to grow and develop. Remember, totipotent is equal to total, everything, okay? Summation of everything. I can be whatever I want to be, none of your business, yeah? So embryonic cells that develop from totipotent stem cells are precursor to the fundamental tissue layers of the embryo are classified as pluripotent, okay? Fundamental tissue layers of the embryo classified as pluripotent, okay? A pluripotent stem cell is one that has potential to differentiation into any type of human tissue but cannot support the full development of an organism. So it's restricted to a human tissue, okay? Whereas, as you can see, totipotent can be any, okay? The cells then become slightly more specialized, referred to as multipotent cell. A multipotent cell stem cell has the potential to differentiate into different types of cells within a given cell lineage or small number of lineages as a red blood cell or a white blood cell, okay? So from tissue, we went to cells, right? So that's multipotent. Pluripotent is related to tissue only, okay? Restricted to tissue, multipotent is restricted to cell. RBC cells, RBC, okay? WBC can be WBC, etc., etc. Multipotent cells can become further specialized into oligopotent cells, and oligopotent stem cells is limited to becoming one of a few different cell types. In contrast, a unipotent cell is fully specialized, can only reproduce to generate more of its own specific type. Stem cells are unique in that way. They can also continually divide and regenerate new stem cell instead of further specializing. Adult bone marrow has three distinct types of stem cells: hematopoietic stem cells, which give rise to RBC, WBC, and platelets; endothelial stem cells, which give rise to endothelial cell types that line blood and lymph vessels; and mesenchymal stem cells, which give rise to different types of muscle cells. All right. So adult bone marrow has three distinct types of stem cells: hematopoietic stem cell, right? Hematopoietic, right? Hemat is blood, which give rise to RBC, WBC, and platelets, right? Endothelial stem cells, which give rise to endothelial cell types that line blood and lymph vessels, right? And mesenchymal stem cells, which give rise to different types of muscle cells. Hematopoiesis. The process of hematopoiesis involves differentiation of multipotent cells, right? We just said multipotent cells are restricted to a particular tissue into blood and immune cells. Multipotent hematopoietic stem cells give rise to many different cell types, including cells of the immune system and the RBC. Yeah, so we have multipotent hematopoietic stem cell. Okay, hemocytoblast. All right, common myeloid progenitors. Remember, progenitors are not differentiated yet. Common lymphoid progenitor. So natural killer cell, large granular lympho, small lympho, T lympho, B lympho, and plasma cell. Common myeloid progenitor, we have megakaryocytes into thrombocytes. Thrombocytes have an active protein called thrombin. Or erythrocyte, mast cell, myeloblast. We have basal, neutral, eosinophil, and monocyte. When you study histology, you'll study all of these, yeah? All right, so as you can see, this is neutrophil, this is eosinophil, this is monocyte, just one. This is basal with all the granules. All right, this is eosinophil, it has a cleavage, remember this? Neutrophils, bundles, as you can see, the 1, 2, 3, 4. All right, macrophage, and we have dendritic cell, okay? Cell fate determination, what is that, okay? Cell fate determination is under the tight control of complex network of transcription factors, right? Lineage reprogramming can be elicited by alternating the expression of levels of these critical genes. Force expression of one or several developmentally important regulatory genes and or maturation factors is commonly used for cell reprogramming. Deleting or suppressing factors that support the original epigenetic state of host to cell facilitate phenotypic conversion, right? Different, there's one's that's genotype, one's phenotype, yeah? Different gene manipulation and the developmental distance between the original cells and the target cells influence the conversion of efficiency and maturation of reprogrammed cells. So cell fate determination under the tight control of complex network of transcription factors, different gene manipulation, and the development distance between the original cells and the target cells influence the conversion efficiency and maturation of reprogrammed cells. So how close they are, how far they are, where are the target cells located, determines the cell's fate. What is asymmetric cell division? What makes it different from a symmetric cell division? The stem cells divide in two ways, symmetry and asymmetry. In symmetric cell division, the two daughter cells will have the same development fate. In asymmetric cell division, the two daughter cells will have different development fates, where one of them will differentiate into a specific lineage. Other cell may continue to remain as a stem cell that divide asymmetrically. Yeah? Symmetric cell division is primarily for proliferation, whereas asymmetric cell division is mainly for cellular diversity. So symmetry is proliferation, growth. Asymmetry is cell division, is mainly for cellular diversity. Differentiate, cellular, and that's why it's asymmetric, grows in multiple different forms. Okay, so symmetric cell division, stem cell, right? As you can see, stem cell, we have two cleavage formation, two cell formation, yeah. Symmetric cell division, yeah. Nucleus, cytoplasmic cell fate determinants. All right, we can actually see cytoplasmic cell fate. These are sister chromatids, right? Microtubules, this particular ones are microtubules. Stem cells, as you can see, stem cell and a differentiating cell. Okay, so asymmetric cell division, this is symmetric cell division. Cell-to-cell communication, very important. This process by which cells interact with each other by sending and receiving signals, typically through chemical messengers called ligands that bind to specific receptors on the cell membrane, allowing cells to coordinate their functions within a multicellular organism. This communication can occur through direct contact or over short distances via secreted signaling molecules depending on the type of interaction involved. Cells communicate, what is the mechanism? Each and every action actually has a mechanism, all right? If you understand the mechanism, if you're in a structure, you'll understand the function, yeah? Cells communicate by releasing signaling molecules that bind to specific receptor on the surface of target cell. Okay, so if you have a target person, what you have to do is, find their email address, send an email, right? So in their inbox, there will be a reception that will happen, yeah? Triggering chain of events inside the receiving cell leading to a specific response, while other to guide a cell fate and tissue formation. You send someone an email, they're going to respond back to you, or decide not to respond back to you. Yeah, so there's actually a specific response that's guided here, okay, which decides the cell fate, okay? You want to take an action, you don't want to take an action, All right, you want to do something, you don't want to do something, okay? And tissue formation, all right? You send an email with an application acceptance. Either they accept it or they deny it. If they accept it, and you go to college. There you go. There's tissue formation. From a cell to you become a tissue, right? There you go. Types of cell signaling: paracrine signaling, autocrine signaling, endocrine signaling, juxtacrine. Paracrine signals act on nearby cells. Autocrine, a cell signals itself. Okay, autocrine, a cell signal itself. Endocrine is hormones travel long distance through bloodstream to reach target cells. Yeah, juxtacrine signaling, direct cell-to-cell contact is required for signaling. Let's say paracrine signaling. You live very close to a college. You want to study at a college. Yeah, you send an application there. You get accepted. You start your college. Your autocrine cell signals itself. Okay, maybe I don't want to go to school. I just want to study myself. I want to be a homeschool person. Something like that. Endocrine hormones travel long distance. You know, I'm a hormone. You know, I want to travel long distance. Let's say you're in Texas. You're going to California. Because you got accepted in a college in California. Okay, juxtacrine, direct cell-to-cell contact is required for signal. There has to be a contact. You know? For example, if you want to get accepted, you better come here and interview. So come here, talk to us, and we'll think about it. Something like that. Key components, ligands. Very important. Ligand, signaling molecule that binds to the receptor. Receptor protein on the cell membrane that receives the signal. You send an email. Yeah. For example, if you want to study at a med school, you have to apply through MCAS, yeah? So, and that's the platform, you have a ligand, you have to apply. There will be someone on the other side of the college that will receive your application, that is a receptor. Signal transduction pathway, the series of intracellular events that occur after the ligand binds to the receptor leading to a cellular response. You send an application, the application was received, they're looking at it, okay? Signal transduction pathway, so you use the signal, right? What did you do? You apply it to the college, yeah? Examples of cell-to-cell communication: neurons communicating at a synapse. Neurotransmitters are released from one neuron and bind to the receptor on another neuron, allowing for nerve impulse transmission. All right, so neurotransmitter released from one neuron and bind receptor to another neuron, allowing the nerve impulse transmission, right? There's a transmission you applied to a college, and then someone received it, allowing for nerve impulse transmission, allowing for information to be transmuted. Yeah, you send your application, that is information, isn't it? Okay. Immune cell communication. WBC interact with each other to coordinate immune response. WBCs are interacting with each other to coordinate immune response. Yeah, so we're coordinating, we're working it out. Development and differentiation. Cells in an embryo communicate with each other. Development and differentiation. Someone's coming to you and asking you, would you be interested in primary care? Would you be interested in something that's more specialized? What is it that you want to do? Talk to us. Come here, okay? Autocrine signal is cell targets itself. I'm targeting myself. Signaling and targeting self. I'm communicating with myself. More like a self-talk, yeah? Paracrine signaling, a cell signals a nearby cell. All right, so a target cell, we have signaling cell. All right, endocrine signaling, a cell targets distant cell through a bloodstream. There's a bloodstream, okay? You're targeting through a bloodstream, yeah? A direct signal, juxtacrine, a cell targets a neighboring cell through a gap junction. Okay, so there's a target cell, there's a gap junction, there's a signaling cell, okay? A direct cell can be considered as a shortcut. Okay, come here, let's connect to a tube. All right, endocrine is usually a bloodstream, yeah? And paracrine is someone that's nearby, para, near. Okay? Remember the word paramedic, very close to the real medicine, yeah? Okay, inducers, cell-to-cell communication inducers refers to molecules or substances that trigger communication between cells by acting as signaling molecules, essentially initiating a cellular response when they bind to a specific receptor on another cell. Example include hormones, neurotransmitter, growth factors, cytokines, small signaling molecules like nitric oxide, which are released by one cell to influence the behavior of the neighboring cell. You know, it's like, you know, you want an attention of a certain individual. You're either saying hi, or you're giving them a smile, or, you know, you're poking them, you're playing loud music to get a response from somebody. There you go. That itself is the growth factor. It itself is a neurotransmitter. Itself is a hormone. Okay? You saying hi with a smile should get a response. That action itself works as a growth factor. All right? They act as chemical messengers to transmit information between cells, coordinating cellular activities within an organism. Binding to receptors. These signal molecules bind to a specific receptor on the target cell surface or inside the cell, which then triggers a cascade of intracellular events. Binding to receptors. You said hi to somebody. There's a growth factor. There you go. The person said hi back to you. And you ask, what is your name? Okay, well, my name is John Doe. Okay. Oh, hi, John Doe. My name is Emily. What's up? You know, how are you doing? How's your college? Are you having fun? Is it too difficult? Would you like to go for lunch? Okay, there you go. And then you know, 5 weeks later, John Doe and Emily are together. Okay, it's actually not a difficult process. Okay, inducers, hormones, long-distance signaling molecules released in the bloodstream to reach distinct target cells, right? Neurotransmitters, chemical released by neurons, the synapses, to communicate with other neurons or muscle cells. Growth factors, proteins that stimulate cell growth, differentiation and survival. Cytokines, small proteins primarily involved in immune system regulation, right? Immune system regulation. Cytokines are extremely important for those that may have cancer. Growth factors, very important. Proteins that stimulate cell growth, differentiation, survival. Okay. Try eating eggs for 10 days and tell me you don't feel better. Okay. Nitric oxide, a small gas molecule that acts as a signaling molecule in various body functions. Very important. Okay. Hormones, extremely important. Try living without hormone, oxytocin, serotonin. Try living without them. Trust me, it is next to impossible. Yeah, neurotransmitters. Try not having perfect neurotransmission. Okay. Trust me, that's not how it works. Chemicals released by neurons at synapse to communicate with other neurons or muscle cells. Very important. Cell migration is the movement of cells in response to a chemical or mechanical signals. Well, we received a signal. There has to be cell migration that's happening. It's a fundamental process that occurs throughout life from embryonic development to tissue repair in adulthood. Right? Why is cell migration important? Development. Cell migration is essential formation of tissues and organs during embryonic development. Tissue repair. Cell migration replaces damaged or old cells with new cells, which helps maintain tissue integrity. Immune response. Cell migration helps the body fight injections by moving phagocytic cells to infected tissues. Okay, so cell migration helps the body fight infections. For example, many people during the storm, right? What they do is migrate to a different state. We just had California fires, right? Many people actually relocated, migrated to different parts of the world, different part of the United States, as a response, okay, to whatever development that was happening in their life. They said, we don't want to, in face of any harm, certainly not fire, certainly not our family, who would like to migrate to a different place. Why? Because we want to fight the economical crisis, the family crisis, the psychological crisis. We want to make sure we're moving our phagocytic cells. We want to make sure we're moving certain things into infected tissues, infected life that we have, the bodily pain that we have. We want to move somewhere else. So that's cell migration. Yes. So there was an action, right? We had certain type of situation that occurred in our life where we had to migrate to a different location. Okay. All right, so these are substrate tractions, this viscous drag from cultural medium, direction of migration, propulsional traction is from, this itself is a migrating cell, okay? We have frictional tractions, rare, okay? The phagocytic cells are very important. Imagine having a wound that's not healing. Our skin is a very important part of our body because it is the first defense against everything. Imagine not having a skin. Your body's going to stick on the walls. You can't even walk without. Imagine having to walk without no soul. You're just walking with all your plantar, organs sticking to the carpets, to the flooring, to dust, mud. You're not going to survive for too long. Okay, how does cell migration work? Signal transduction cells receive signals that activate a series complex of cascades. You got an alert, weather alerts. Oh my God, there's fire, move out. Cytoskeletal remodeling. The cytoskeleton changes shape in response to signals. Cell to substrate interaction, cells adhere to each other and to the extracellular matrix, ECM. Matrix metalloproteinases, MMPs. Cells produce MMPs to create gaps in the ECM, allowing them to pass through. What can go wrong with cell migration? Cells may not migrate at all. Cells may migrate to the wrong location, right? These issues can lead to abnormalities or life-threatening. You may decide, well, I understand it's fire, but this is my place, and I'm not going to move, okay? Second, you may move to another location where you're going from a pan to a fire. You're going from fire to a fire, all right? You're going from the east direction of the mountains to the west direction of mountains, where there are chances, there are trees, and there might be another fire, right? Third, these issues can lead to abnormalities or life-threatening conditions, okay? You move at a certain location, but now that itself is life-threatening. Why? Because you may not be okay with the cold temp. You're going from the hot weather of California to something that's freezing cold, okay? There you go. Moving to Alaska mountains, from one mountain to another, maybe because you like maybe because you like mountains, but then again, from fire, you're going to freezing cold. Your body's not used to surviving, There you go. So these issues can lead to abnormalities or life-threatening condition. All right, cell death is process by which a cell stops functioning and dies. Yeah? That's a natural process that removes damaged or unnecessary cells to maintain tissue homeostasis, right? Homeostasis is the basic necessity of the human body, okay? You can't have a very low pH, etc. Programmed cell death, a genetically programmed process that eliminates damaged or infected cells. It's programmed, Once you're no longer useful to us, there you go, we're done. Accidental cell death, an uncontrolled process that caused by injury, disease, or exposure to toxin. Yeah, it happens, right? Your tissue can die. Yeah. Types of cell death: necrosis, pyroptosis, necroptosis, apoptosis. Okay, different types of death. An unprogrammed process that occurs when a cell lacks blood supply or is exposed to toxins, okay? Cell lacks blood supply. Try bleaching your hair, okay? Or try bleaching a part of your skin, okay? Or try having a wound, and not healing it, okay? But if you really want to see something that smells necrotic, try to smell a gas gangrene. You understand what necrosis is, okay? Pyroptosis, a programmed process that occurs in cells infected with certain viruses or bacteria. Yeah, necroptosis, a programmed process that's like necrosis, but involves specific proteins in the dying cell. Necroptosis is a little specific to a certain protein. Pyroptosis is a program infected with viruses or bacteria. Necrosis is unprogrammed. Yeah. Apoptosis, apoptosis is the process of programmed cell death. It is used during early development to eliminate unwanted cells. For example, those between the fingers of a developing hand. Okay, in adults, apoptosis is used to rid the body of cells that have been damaged beyond repair. Apoptosis also plays a role in preventing cancer. It's programmed cell death. Imagine having accumulation of dead cells in your body. Okay. All right. So apoptosis, normal cells, formation of blabbing. The cells breaks apart into several apoptotic bodies, which are then phagocytized. No inflammation is caused. No inflammation. There is no reaction. Yeah, they just die. That's it. From a normal cell, necrosis, cell swells. And that's exactly why I said, if you want to see necrotic cell, you have to see a wound where no healing was done. You can actually smell the stench of necrotic cell, okay? Plasma membrane rupture, cellular and nuclear lysis cause inflammation, and that's what causes the smell, okay? Cell death benefits cell that helps regulate cell numbers, yeah? It removes damaged or infected cells, which can help prevent spread of pathogens, yeah? It supports morphogenesis during development. Problems can arise when cell death doesn't happen as expected, or cells die when they should not. Healthy cell dying is a problem, yeah? Impaired or excessive cell death can promote disease. Okay, impaired or excessive cell death. Too much of cells dying is also not good. Impaired or you're impairing, you're stopping a cell death can promote disease. Yeah. Cell regeneration is the process of something's dying, something has to regenerate. Yeah. Cell regeneration is the process of restoring, replacing damaged or missing cell tissues and organs. Now, cell death is important. Cell regeneration is also equally important. It's a natural process that helps organisms maintain their tissues and organs. Okay. How does it work? Cell differentiation. We saw molecular signaling. We saw macrophages. Yeah? Cells that have already developed into their final form can revert into stem cells. A complex network of molecular signaling governs the regeneration process. Macrophages. These cells are first to sense injury, releasing cytokines and chemokines, that stimulate other cells to enter the cell cycle. As you can see, we have growth, cell division. Two cells are formed. This is regeneration. Yeah. Okay, senescence and age. Senescence is a process that occurs as a part of aging. It's characterized by a gradual decline in an organism's functional abilities. Cellular senescence is a hallmark of aging that occurs when cells exit the cell cycle in response to stress or damage. What happens during senescence? Cell cycle exit. Senescent cells stop dividing and are unable to proliferate. Senescence-associated secretory phenotype, SASP, senescent cell release a variety of proteins, chemokines, growth factors, and proteases. Right? Tissue disruption. Senescent cells can disrupt tissue homeostasis and normal tissue structure and function. Inflammation. Senescent cells can cause local and systematic inflammation. How does senescence affect the body? Impaired immune system. As people age, their immune system becomes less efficient, which allows senescent cells to accumulate. Cognitive decline. Senescent cells in the brain can impair cognitive function. Skin changes. Senescent cells in the skin can cause wrinkles, sagging, and thinning. Yeah. What diseases are associated with senescence? Cancer, diabetes, osteoporosis, cardiovascular disease, stroke, Alzheimer's disease, and osteoarthritis. Fetal circulation. Fetal circulation system of blood flow in a fetus, which includes a heart, placenta, and umbilical cord. The fetus circulation system uses shunts to bypass the lungs and liver, which aren't fully functional until birth. How it works. Oxygenated blood from the mother enters the fetus right atrium through the placenta. Most of the blood flows through the foramen oval to the left atrium. The blood then moves to the left ventricle and into the aorta. The blood is sent to the brain, heart, arms, and lower body. The blood returns in the right atrium to the superior vena cava. Most of the blood flows through the ductus venosus to the inferior vena cava. The blood then enters the umbilical arteries and flows to the placenta. The placenta exchanges oxygen and nutrients with the mother's blood. The blood returns to the fetus through the umbilical cord. Yeah. So as you can see, the blood's actually the fetus through the umbilical cord. But as you can see, the oxygenated blood from the mother enters the fetus right atrium. Okay. Okay. So this is actually the flow, or this is the placenta. This is the umbilical cord, right? As you can see, this umbilical vein. All right. Now this is mother's fully grown body, and this is placenta. Okay. So as you can see, what is going on, right? Ductus arteriosus, pulmonary artery, lung, left kidney. We have umbilical arteries. We have liver. We have ductus venosus. We have lungs. We have foramen oval. We have aorta right here. So basically, the blood is flowing, and the blood is flowing back to the mother's heart. Okay. Pulmonary trunk, lungs, pulmonary veins, left atrium. All right. So this is umbilical vein, this is placenta, right? Umbilical vein, ductus venosus, inferior vena cava, right atrium, right ventricle, pulmonary trunk, lungs, pulmonary veins, left ventricle, ductus artery, aorta, systematic circulation, umbilical arteries, and back to placenta. Then again, the blood's flowing back, okay? Lungs, pulmonary veins, left atrium, okay? Because the organs in a fetus are not fully developed as of yet, so it survives on mothers. That includes the blood supply and also the drainage system. Gestation and birth. Human gestation lasts for 280 days, equivalent to 40 weeks calculated from the first day of the last menstrual period. First trimester, the first time of pregnancy, the first 3 months of the weeks 1 to 13 of pregnancy. It is a time significant change for the mother and the baby. Baby development. What happens in the first trimester? The baby's major organs, body structure form, including heart, brain, gonads, spinal cord, digestive system, eyes, ears, liver, and limbs. Miscarriages and birth defects. Most miscarriages and birth defects occur during the first trimester. By 5th week, it's 10 millimeter in length, and 6th week, it's 15 millimeter in length. End of 8th week, most organs are formed, and the brain is developed, and the embryo becomes known as fetus. The second trimester, week 13 to week 27, the second trimester of pregnancy is often thought of as the best part of pregnancy. Fetus goes through tremendous growth, starts moving in the amniotic fluid. The fetus will move and even do flips throughout your second trimester. The first movement is called quickening. The first is quickening. End of 6 months, the fetus 30 to 36 centimeter long. It is a time when many early pregnancy symptoms ease and you may feel more energetic. Okay, so fetus continues developing during this trimester, adding weight and taking on the features of a newborn. Your care provider can determine the sex of the fetus on an ultrasound, often around 20 weeks. Third trimester, fetus continues to grow and weight. So weight increase, it also grows. The lungs are still maturing and the fetus begins to position itself head down. By the end of third trimester, the fetus is about 19 to 21 inches long, so weights on average 6 to 9 pounds. The fetus can see and hear, the brain continues to develop, and the kidneys and the lungs continue to mature. By the 36th week, the head may engage, drop into the pelvic area, a process called as lightening. The bones of the skull remain soft to make it easier to pass through the birth canals. For many babies, the irises of the eyes are slate blue. The permanent eye color will not appear until several days of week after birth. The fetus can suck its thumb and cry. By 38 to 40 weeks, the fetus, lanugo, the fine soft hair on the body and limbs, has disappeared almost completely. By 38 to 40 weeks, the lungs have matured completely. Okay, initially we didn't have mature lungs. That's why it was dependent on the mother. Okay? The baby is covered in vernix caseosa, or simply called vernix, a creamy protective coating on the skin. Yeah, we all have seen. It's not just human, it's also animals. If you watch National Geographic, you'll understand it better. The head will usually turn downward during the last couple of weeks of pregnancy. Why? Because it's ready to come out. Antibodies are transported through highly selective active transport from the mother to the fetus, right? Antibodies are transported through highly selective active transport, antibodies that are needed for the baby. This is exactly why a baby's immunity during the first few weeks is fully dependent on mother's milk, okay, right? And that's why it's very important for a baby to drink mother's milk, because they have higher antibodies and it helps their immune system, okay? There are many types of pregnancy, including intrauterine, ectopic, molar, and multiple pregnancies. What is intrauterine pregnancy? This is where embryo is, okay? Ectopic pregnancy this is where embryo is. It did not line itself in the endometrium, yeah? Intrauterine pregnancy, a normal pregnancy where a fertilized egg attaches to the lining of the uterus. Ectopic pregnancy, a fertilized egg attaches to a structure outside of the uterus, such as a fallopian tube, cervix, or abdominal cavity. Fallopian tubes, that's where the fertilization occurs, right? For the fertilized egg to be a fully grown embryo, it has to attach itself to the uterine wall. Yeah, ectopic pregnancy can be life-threatening and are usually not safe to continue. About 1% of pregnancies are ectopic. All right, ectopic implantation, what is it? The concept may be arrested at any point. Conceptus can be arrested at any point during its migration through the uterine tube and implant in its wall. It got arrested. It did not complete the full cycle. Previous pelvic inflammation damages the tubal epithelium, may predispose to such delay in tubal support. Your fallopian tube is not functioning for several reasons, and that's why it did not occur. This is exactly why mothers are said, don't drink, don't smoke, etc., etc. Presence of an intrauterine contraceptive device or the use of progesterone-based oral contraceptive may also predispose to ectopic pregnancy, probably because of alteration in the normal tubal support transport mechanisms. Right, so this is, as you can see, this is caesarean scar, or abdominal, right? Cornual, this is isthmic, this is ampullary, this is fimbrial, okay? Abnormal implantation within uterus. The implantation near the internal results in the condition of placenta previa with its attendant risk of severe antepartum hemorrhage. Right? This is complete, this is partial, this is marginal, and this is low lying. Molar pregnancy, a non-cancerous tumor that occurs when an egg and sperm fertilize incorrectly. Symptoms include vaginal bleeding, nausea, vomiting, and preeclampsia. All right. So as you can see here, fluid-filled cyst tumors, that is molar pregnancy. If you have so much tumor, there is actually a space to develop. Yeah. Multiple pregnancy carrying more than one baby at a time, such as twins, triplets, quadruplets. We've seen octuplets, right? We've heard the story of octomom. This is dichorionic, 2 placentas. This is diamniotic, 2 sex. Monoamniotic, 1 sex. Okay, so this is placenta previa, normal placenta, marginal, this is partial, and this is total. Okay, in placenta previa, the placenta attaches low in the uterus. The placenta might partially or completely cover the opening of the uterus called the cervix. Okay. The placenta previa can cause severe bleeding in the mother before, during, or after delivery. The placenta is an organ that develops inside the uterus during pregnancy. Twining. In embryology, the process by which 2 embryos develop from a single fertilized egg or 2 fertilized eggs. The type of twining depends on how early in development the fertilized egg splits. Identical twins, also known as monozygotic twins, identical twins develop when a single fertilized egg splits into 2 embryos. Identical twins share the genetic information. Identical twins can be monochorionic, monoamniotic, monochorionic diamniotic, or dichorionic diamniotic, mono. Also known as dizygotic twins. Fraternal twins are also called as dizygotic twins. Fraternal twins develop when 2 separate eggs are fertilized by 2 separate sperm. Okay. Fraternal twins have different genetic information and are not more or less alike than regular siblings. So they're not identical. They're fraternal twins, depending on who the different fathers. Conjoined twins. Conjoined twins are are a type of identical twin that separate after day 10 of development, resulting in fetuses that are attached to each other. Right? You've heard stories, you watch YouTube videos of conjoined twins. Okay. Twining. Morula, right? We just said 36-ish. Above is morula. Cleavage days 1-3, dichorionic diamniotic. Okay. Cleavage days 4-8, monochorionic diamniotic. Okay. As you can see, these are different placenta. That here is the same placenta. Alright, implanted blastocyst, cleavage days 8-13, monochorionic, monoamniotic, formed embryonic disc, cleavage, disc 13-15, conjoined twins. So these are conjoined twins, these are monoamniotic, they're different. As you can see here, they are conjoined. Before pregnancy, that's HCG levels. Embryo implantation, okay, and embryo loss. Chemical pregnancy, a pregnancy that occurs before an ultrasound scan, usually 4 to 6 weeks into a pregnancy. The body produces hormone called as HCG, but the pregnancy is short-lived. A chemical pregnancy or a biochemical pregnancy is a very early miscarriage that happens within the first 5 weeks of pregnancy before the pregnancy can be seen on an ultrasound. An embryo forms and implants in your uterine lining, but then it stops developing. Chemical pregnancies occur so early that many people don't realize they have miscarried. Yeah, Chemical pregnancies get their name from the hormones that produce a positive result on a pregnancy test. All right, so that's it for human embryology. We will be talking, in detail about human embryology in the future videos. Okay, so if you like this video, please subscribe to my channel, like this video, share it with your friends, your co-students, and comment and tell me if you really like this video and if you want me to post more videos on human embryology. Okay, bye.